Can somebody confirm you can hear me?
I can hear you.
Thank you. Four. Before we begin, I'm going to talk a little bit about the grading. I've got Lab 4 graded, and some of the extra credit. Let me go to the grade book. There's one extra credit I have not graded. There are two that I have graded. Um, what else do I have graded? Something else is graded too. So take a look at your assignments and what's been graded. Lab 5 has not been graded, and so that will probably be postponed because I've got to grade the Tuesday, Thursday people, their Lab 4. So I doubt that I'm going to get to Lab 5 until later this week, but we'll see. Let me go to the grade book myself, then I'll have an easier time talking about this. I'm not sharing my screen, so I can open up the grade book and see. Oh, I graded the extra credit study sheet. Student discussions were graded. I've now reopened that assignment for students to discuss in it. However, you will not get extra credit point. But it is now open so you can reuse the student discussions. I have extra credit study 2 graded. Extra credit study 3 is not graded, but extra credit study 4 is. I'll come back to grading extra study 3. I have not graded any of the labs yet for the second time, but I have graded lab 00, lab 01, lab 2, lab 3, and lab 4 for the first time. On lab 4, if you did not get 100%, which many of you did not, you can resubmit it by February 10th for full credit. A lot of you are having a hard time with the lab, and I do make myself available to helping out your lab work. And some of you should really take advantage of my ability to help you during the lab. So let me encourage it. I think that's it for the grades. Like I said, lab 5, let me change that. I'll make it so that the until date is not going to be this Saturday. I have a feeling it'll be a week from this Saturday. I guess I don't really know until I've got it graded, but I'm pretty sure I'm not going to get it graded early this week. Oh, where the heck do I go to change this? Edit. All right, any question about the grading? If not, let me get out of here. Close down the grade book, I mean. And let me share my screen now. So you should be seeing my screen. Today is February 5th. Boy, that's really late in February already. And let me remind you, you have a quiz this Wednesday, which will cover labs 3 and 4, chapter 3 in the end of chapter 2. So that's what the quiz will cover this Wednesday. Today, it looks like we're supposed to be starting chapter 4. Did we finish chapter 3? Can somebody confirm that? Let me take a look.
Yeah, we finished chapter 3.
Did we start chapter 4?
Yeah, we did.
All right, I don't have that marked here on chapter 3, but let me check a look at chapter 4. Oh yeah, we had talked about this slide, comparing gram-positive cell walls and gram-negative cell walls last time. So there are differences between the bacteria and their cell walls. The gram-positive cell wall is a thick cell wall with a thick peptidoglycan layer. The gram-negative cell walls do have peptidoglycan in them, but they are much thinner cell wall. And I should put a caveat that there are some bacteria that do not have cell walls, and they will stain as gram-negative. But majority of them have a thin bacterial cell wall with a thin layer of peptidoglycan. And those gram-negative cells have an outer membrane that does contain lipid A in the cell wall. All right, let's talk a little bit about the gram stain mechanism. The crystal violet iodine complex forms in, well, essentially all bacteria cells. In the gram-positive cells, the crystal violet iodine complex does not leave the thick peptidoglycan. In the gram-negative cells, the alcohol will dissolve the outer membrane of the cell wall, and then the crystal violet iodine will wash out of the thinner peptidoglycan layer. Safranin can then get into the cells and stains the cells pink. And that's the difference of the gram stain, why they stain differently. This table is showing you a summary. Let me blow this up, of the gram-positive cells and the gram-negative cells. Obviously, the gram-positive cells retain the crystal violet complex, and the gram-negative cells do not retain the crystal violet iodine complex. The peptidoglycan layer is thick in the gram-positive, thin in the gram-negative. Teichoic acid is present in the gram-positive cells. It's essentially absent in the gram-negative cells. The periplasmic space is absent in the gram-positive cells, present in the gram-negative cells. The outer membrane is absent in the gram-positive cells, and it is present in the gram-negative cells. There's a lipopolysaccharide containing lipid A. It's virtually none in the gram-positive cells, so I'm just going to say none. And it's at high concentration in the gram-negative cells. There is a mistake in this table, and it says the peptidoglycan layer is a thin layer. That's correct, but then it has single layered. And like I said, the minimum is like 3 layers of peptidoglycan. And it's something to like 20 to 30 layers on the high side. Any question about this table? You can ignore the lower portion of the table. There are some differences in the cell walls of prokaryotes. First, let me talk about the archaea. And I should move that up because I always start about this one first. And that is the archaea are not bacteria. So it's not surprising that they have differences from the bacteria. They belong to a different domain and they have different cell walls. Their cell walls are made of pseudomurine instead of peptidoglycan, and pseudomurine is another disaccharide similar to peptidoglycan. And some of the archaea lack cell walls, and obviously there's a few bacteria that lack cell walls too. So the archaea differ in their cell walls. It's curious that the archaea, when we stain them with the gram stain, they have two different cell types. They have the gram-positive cells and they have gram-negative cells. And of course, well, I don't know, of course, to me, of course, but the reason why the archaea are gram-positive is because they have a thicker cell wall with obviously a thicker pseudomurine layer. And then the gram-negative cells have a thinner cell wall. And so the archaea do stain similar in the gram stain. It's for similar reasons too, the thickness of the cell wall. In the bacteria, we have two different families that are atypical in the cell walls of bacteria. And these are the family of Mycoplasma and the family of Mycobacterium. In the family of Mycoplasmas, this family lacks a cell wall. These are the bacteria that lack a cell wall. And it's curious that the Mycoplasmas have sterols in their plasma membrane. And the sterols give strength and support to the plasma membrane, very similar to the way animal cells have cholesterol in their cell membranes, which give our cell membranes strength and support. So the sterol is acting the same way as cholesterol in our cells. The sterols in mycoplasmas, I should say. And that's because these cells don't have a cell wall, and so they need a little stronger cell membrane to help hold the cell together. The Mycobacterium are another family of bacteria that have atypical cell walls, and they have macolic acid in their cell walls. And let me show you a little picture of that. I need to have it in this lesson and I don't, so I'm just going to go to Google. Macolic acid in cell wall. Look at the images. Let's take that one. So here is the cell wall of the Mycobacterium. And you can see they have a normal cell membrane. And they do have peptidoglycan. The Mycobacteria are gram-positive cells. But then they have another layer of something I'm not going to talk about, and then a very thick layer of mycolic acid. It is actually a little thicker than the peptidoglycan layer. Mycolic acid is a lipid, and that makes these cells impenetrable by water-soluble agents. Something that wants to come into the cell has to come through the mycolic acid, and this is very good at keeping out water-soluble molecules, such as crystal violet and safranin of the gram stain. The mycolic acid also is very good at keeping water that's inside the cell from leaving the cell, and that's probably the main purpose for mycolic acid, that the mycobacterium can survive harsh environmental conditions like dry conditions, but most bacteria cannot. Obviously, the endospore can survive that, but the mycobacterium can survive harsh, dry conditions because of the mycolic acid in the cell wall. And it also keeps water-soluble molecules out of the cell. And crystal violet and safranin are water-soluble. And that's why these cells, they stain poorly with the gram stain. They do stain, and they do stain gram positively if you're doing the gram stain correctly. It's just that they're lighter in their stain. Any question about any of that? Let's see where we are. So the mycobacterium are gram-positive cells, but they have mycolic acid in them. That mycolic acid makes them an acid-fast staining cell, meaning if we use a dye that's an acid-fast dye, it will get in the mycolic acid and stay in the cells. Whereas most bacteria, you stain them with the acid-fast stain, and it will decolorize out of the cell. But the mycolic acid keeps the acid-fast stain in the cell, and that's why we call the mycobacterium an acid-fast staining cell. Any question about the mycobacterium or the mycoplasmas? The mycoplasmas, by the way, stain gram-negatively because they don't have a cell wall to retain the crystal violet iodine complex. The mycobacterium do stain gram-positively, but they're atypical gram-positive cell. You can damage the prokaryote cell wall, such as putting the enzyme lysozyme on them that will digest the disaccharide and the peptidoglycan. You can also damage the cell wall by applying penicillin G. This antibiotic will inhibit the peptide bridges and peptidoglycan, so the cell wall will have holes in it, and then the cell membrane can protrude out of the hole. You don't need to know the terms for the wall-less bacteria, but you can damage the prokaryote cell wall with either lysozyme or penicillin. And that's actually how penicillin G kills the cells, by damaging the cell wall, and then water will move into the cell. The cell membrane will then protrude out of the holes in the cell wall. More water will move into the cell until eventually the cell membrane will pop from extending too far outside of the hole of the cell wall. So the gram-negative bacteria are not as susceptible as the gram-positive to some agents, such as penicillin G and other antibiotics, like the cephalosporins, or at least some of the cephalosporins. That doesn't mean the gram-positives are not sensitive to antibiotics. For example, not methicillin, which that does kill gram-positive cells, but ampicillin, one of the early members of the penicillin family to be discovered, actually worked on both gram-positive and gram-negative. Penicillin G only works on gram-positive cells. And then there are other antibiotics besides most of the later members of the penicillin family can work on the gram-positive cells. But there's also streptomycin, which will work on the gram-negative cells because it's a lipid-soluble antibiotic, and so it'll go right through the outer cell membrane of the gram-negative cells. Any question about any of that? So the outer membrane can protect the gram-negative cells from a few agents that will do damage to the gram-positive cells. And we'll talk a little bit about the gram-negative and gram-positive cell in just a little bit. Before I talk about that, I want to talk about the plasma membrane. It is mostly a phospholipid bilayer, and the phospholipids arrange themselves in this direction, where inside the cell, the heads containing the phosphate group will be facing the cytoplasm because the phosphate groups like to be around water of the cytoplasm. The phospholipid tails will be on the inside because they don't like to be around water. And then outside the cell will be the phosphate heads once again, because outside the cell will be mostly water. You can think of this as a red blood cell. Inside the blood is mostly water. So the phosphate heads will be facing the water. And it doesn't matter what cell you are, most organisms are mostly water. So outside the cell, there is mostly water. The phospholipid bilayer is the main molecule of the plasma membrane, but there are other molecules in the plasma membrane, such as carbohydrates and proteins. There are three types of proteins I want to talk about. There are peripheral proteins, integral proteins, and transmembrane proteins. Peripheral proteins are proteins that are mostly on one side of the membrane or the other. And that's why they're called peripheral proteins. They're peripheral to the cell membrane. The other proteins, the integral proteins and the transmembrane proteins, span the membrane, such as this pore or channel protein. It's a protein that makes a pore in the membrane, and then this protein can allow molecules to either enter the cell or leave the cell. Another protein that spans the membrane is a transport protein. We've talked a little bit about that, and it will move one molecule from outside the cell to inside or vice versa, even against the molecule's concentration gradient. And this requires energy for that transport protein to transport the molecule against its concentration gradient. And that energy will be supplied by ATP. All right, any question about the plasma membrane? The plasma membrane is described using the fluid mosaic model. This model states that the membrane is a mosaic of phospholipids and proteins, as well as other molecules, but mainly phospholipids and proteins, and that this membrane is as fluid as olive oil, at least at body temperature. And that means that the molecules can actually move around in the membrane because it's as fluid as olive oil. And those of you who have taken A&P, you know that many proteins, when they receive a ligand giving the cell a signal, these receptor proteins will move together, come together in the membrane before they're brought into the cell. And that's how the proteins function. They actually move about in the membrane. There are many proteins that do that. Probably the majority of proteins do move about in the membrane. The phospholipid molecules can also move about in the membrane. Not all proteins can move about in the membrane. If the protein is free in the membrane, it'll move about. But if the protein is either anchored to the intracellular matrix, that is a scaffolding of proteins running throughout the cell on the inside of the cell. So it's running throughout the cytoplasm, and it gives a backbone to the cell. If the protein in the membrane is bound to the intracellular matrix, that protein will not move about in the membrane. And if the protein is bound to an extracellular matrix protein, meaning some extracellular content outside the cell, then that protein will not move about and is not free to move about in the membrane. It'll stay in one position. You can think of that maybe like a bone cell that's in the bone. It can have a protein that's actually anchored to the matrix of the bone, and then that protein will not move about in the membrane. But many other proteins move about in the membrane, and sometimes they have to move about for the proteins to function. The phospholipids can also move about in the membrane. They can both spin, meaning spin around this way and stay in this position, and then they can move from this position to that position. Okay, water can actually move through the lipid bilayer of the phospholipid bilayer by simply coming up here, make my mouse alive, then waiting for this phospholipid to move, and then when it moves, it takes this position. And it can't get through this side of the phospholipid membrane, but it just waits right here in the phospholipid bilayer, and it can't close because there's water in there, but it simply waits for this. So let's make it this one here. Come on, mouse. This one here. It simply waits for this one here to move, and then the water can move from outside the cell to inside the cell. And of course, after it moves through, then the phospholipid can move over and close off the lipid bilayer. And once it moves into the cell here, this one can move over and close off the lipid bilayer here too. But water can move through the membrane. It has to wait for the phospholipid to move, and then it can move into the open space, and then has to wait once again for the phospholipid in the lower portion of the, membrane to move before the water can move in. But water can move into the cell. That is not good enough for plants and their roots to absorb water, and especially plants and their roots will have aquaporins. These are transport proteins to move water from one side of the membrane to another. And aquaporins are actually very common proteins in cells. Many cells have aquaporins in them, not just the roots of plant cells, but you can see it. That would function. You can see that would function because the roots are going to want to take up the water, and they have these aquaporins proteins to bring up the water. But many other plants, many other cells also have aquaporins to transport water mainly into the cell. The plasma membrane shows selective permeability. That means it allows the passage of some molecules, but not other molecules. No large molecules can be transported through the plasma membrane because it can't get through the phospholipid bilayer unless there is a transport protein in the membrane to transport that molecule across the membrane, regardless of whether it's transporting it into the cell or out of the cell. A large molecule needs to have a transport protein transport the molecule. And that's true of things like glucose, fructose, all of the amino acids, anything the cell would want, it will generally bring it into the cell with a transport protein. And there will be one transport protein to bring in one specific molecule. meaning there's one transport protein to bring in glucose, a second transport protein to bring in fructose. Ions do not move across the membrane, or if they do move, they move very slowly across the membrane. Lipids, like, I don't know, cholesterol or oil molecule, can move right through the lipid bilayer because the lipid soluble molecules can move through the phospholipid bilayer. However, if you think about it, like a cell in the blood, there are not many lipids in the blood. The blood is mostly water, and it contains mostly water-soluble molecules. But lipid molecules can move through the cell membrane. Small nonpolar molecules like oxygen and carbon dioxide can also move through the cell membrane. And as I mentioned, water can move slowly through the membrane, but it has to wait for the phospholipid to move out of the way, and then it can move into the first lipid bilayer, and then it has to wait for the phospholipid on the other side of the bilayer to move before the water can move into the cell. Oxygen and carbon dioxide are the most important molecules that can move across the membrane, and we need oxygen to move into the membrane and into the cell to keep the cell alive. And oxygen can diffuse right through the cell membrane. And then carbon dioxide can diffuse out of the cell, diffusing right through the cell membrane. Any question about any of that? All right, the plasma membrane and bacteria contains the enzymes for making ATP production. So the enzymes that make ATP in bacteria are actually in the cell membrane. It's also true that of photosynthetic prokaryotes, that the photosynthetic pigments are found in the plasma membrane. And let me see if I can blow this up a little bit. What we have is a folding of the photosynthetic bacteria's cell membrane so that the membrane does not go straight across here's the cell wall. The membrane isn't going straight across here. What it is doing is, here's the membrane here, and then it does a fold coming around. Got this a pointer for some reason. Oh, that's why it's a pointer, because I have it blown up so much. But the point is that the membrane comes like this, and then it makes a fold, and then it goes like that, then makes another fold. And inside these folds, we have the photosynthetic pigments. There is a name for it. You don't really need to know it, but chromatophores or thylakoids. You can damage the plasma membrane. And if you do damage the plasma membrane, you will allow the cytoplasm to leak out of the cell, and that will kill the cell. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and then the polymyxin antibiotics, like polymyxin B, puts a hole in the cell membrane. And then the cytoplasm can leak out of that hole, killing the cell. And that's how polymyxin actually kills at least gram-positive cells. Gram-negative cells, we'll talk about that later. Polymyxin can kill the cell either by putting a hole in the cell membrane, or polymyxin can put a hole in the outer membrane of a gram-negative cell, and that will kill the cell as well. All right, material has to move across the cell membrane in order for the cell to stay alive. The movement of materials across the cell membranes is critical to the cell. Movement can happen by two different processes. Molecules can move passively across the cell membrane, or molecules can move actively across the cell membrane. If the molecule moves passively across the cell membrane, the molecule will move down its concentration gradient, meaning the molecule will move from areas of high concentration to areas of low concentration. For example, in a cell, oxygen is at low concentration in the cell and at higher concentrations outside in the air. And that's why the molecule can passively move into the cell by simple diffusion. Simple diffusion is the movement of a solute molecule down its concentration gradient. Any question about that? Facilitated diffusion is another way that a solute molecule can move down its concentration gradient, moving from areas of high concentration to areas of low concentration. The difference is simple diffusion, that's all there is. In facilitated diffusion, the molecule is moving through a transporter protein across a membrane. So the transporter protein is facilitating the diffusion of this solute molecule through the membrane. And that's why it's called facilitated diffusion. Any question about any of that? All right, the third passive process is osmosis. Osmosis is it does not involve a solute. It's involving the movement of water, and water is not a solute, it is a solvent. So osmosis is a solvent moving across a membrane, meaning water moving across the membrane is osmosis. And in a cell, water generally moves from outside the cell to inside the cell by osmosis. Any question about any of that? Once again, osmosis occurs by the movement of water from areas of high concentration to areas of low concentration. Generally from outside the cell to inside the cell. But if a plant, for example, were to be planted in the desert, the outside the cell will be less water than inside the cell, in which case water will leave the cell by osmosis. Once again, osmosis involves a solvent, not a solute, and the water has to move across a what do you call that? A membrane, either a living membrane like the cell membrane or a artificial membrane. All three of these are passive processes. There's also an active process, and that is where the molecule moves against its concentration gradient from areas of high concentration to areas of low concentration. So the molecule is moving from high concentration to an area of low concentration. How that happens is we have a transport protein that moves the molecule from areas of low concentration to areas of high concentration. This transport protein requires the energy of ATP to move one molecule across the membrane. So one ATP is used up to move one molecule by active transport. And let me state that to move the molecule against its concentration gradient takes work, and that work uses up the energy which is obtained from ATP. Let's talk a bit more about the passive processes. Simple diffusion is where a solute moves from an area of high concentration to an areas of low concentration. Like for example, if we were put a drop of dye in this beaker of water, the dye will diffuse from areas of high concentration to areas of low concentration. And if given enough time, there will not be this blob of dye in there. It'll be entirely light purple, evenly throughout the beaker of water. And that's because the dye is moving by simple diffusion. Simple diffusion can occur in the absence of a membrane, but remember, simple diffusion can occur across a membrane, such as when oxygen diffuses across the cell membrane. And like I said, oxygen and carbon dioxide are the most important molecules to move across the cell membrane by simple diffusion. Facilitated diffusion is where a molecule moves from areas of high concentration to areas of low concentration. The molecule moves across a membrane, such as the cell membrane, using a transporter protein. And this is showing you for glucose. I'm going to tell you, scratch the glucose. A transporter protein is actually how fructose moves into a cell. So I'm going to talk about facilitated diffusion with fructose. So if you eat a lot of fruit, you're going to have a lot of fructose, that sugar in your gut, and it'll be at high concentration outside your gut, and the fructose will be at lower concentration inside the gut. We have a transporter protein, which is like a channel protein to allow the fructose to move from areas of high concentration in your gut to inside your gut cell, where the fructose concentration is lower. So this is facilitated diffusion because there's a protein facilitating the diffusion of fructose from areas of high concentration to areas of low concentration. What this means is you will never digest all of the fructose, because once the fructose concentration outside the cell equals the fructose concentration inside the cell, there will be no net movement of fructose inside the cell, or for that matter, outside the cell. So the fructose, you will never digest it, because once it's the same concentration inside the cell as outside the cell, you don't digest the fructose any further. That's not the case with glucose. Glucose, if it's at high concentration outside the cell and low concentration inside the cell, we have a transporter protein which will facilitate the movement of glucose into the cell. This will not cost the cell any energy because the molecule is moving from areas of high concentration to areas of low concentration through this transporter protein. However, when you eat sugar or any other molecule containing glucose, all of those glucose outside the cell will be digested and brought inside the cell. And that's because this fructose transporter protein, once the glucose concentration outside the cell equals the concentration inside the cell, the protein will change from being a facilitated diffusion protein to an active transport protein. And then this active transport protein will pump every glucose molecule outside the cell to inside the cell until there's zero outside the cell. So when you eat glucose, you will digest all of it. Any question about any of that? Now, you don't really need to know glucose works first as a facilitated diffusion and then switches to being an active transporter protein. You don't really need to know that, but you should know about facilitated diffusion. and how the molecule moves through a protein, through the cell membrane, from areas of high concentration to areas of low concentration. All right, in both simple and facilitated diffusion, it's a solute that's transported, and the molecule only moves down its concentration gradient from areas of high concentration to areas of low concentration. With osmosis, this is not where a solute is transported across the membrane, it's where a solvent, specifically water, is transported across a selectively permeable membrane. And this membrane can be an artificial membrane, or it can be the cell membrane, or like an organelle membrane. The water molecules only move from areas of high concentration to areas of low concentration. So in that sense, osmosis is, I don't know, sort of similar to facilitated diffusion. Sort of similar to diffusion too, but it's by a solvent, not by a solute. And then osmosis is always across a membrane, like facilitated diffusion. Now, it's the water that's moving by osmosis, but when we're talking about the tonicity, you should know that the tonicity is not defined by the concentration of water. The tonicity is defined by the concentration of the solvent, no, solute, sorry, by the solute. Wherever the solute is high in concentration, the water will be low in concentration. And wherever the solute is low in concentration, the water will be high in concentration. And so you can always remember that the solute will be opposite of the solvent in its concentration. And usually it's the water that moves across the membrane, even though the tonicity is defined by the solute concentration. There are three environments that a cell can find itself in considering the tonicity, or three osmotic solutions. You can have an isotonic solution, a hypotonic solution, and a hypertonic solution. You should note that each of these is relative term, meaning it's correct to say that the solution outside the cell was hypertonic relative to the solution inside the cell. So when we're saying these three, we're talking about a relative comparison. The easiest to talk about is isotonic. An isotonic solution is wherein the concentration of the solute outside the cell is the same as the concentration inside the cell. Remember where the solute is, there will be water, and that will mean the water outside the cell is equal to the water inside the cell. The cell will be happy in an isotonic solution because water will not move into the cell or leave the cell. There will be no net movement of water, I should word that. The cell is happy because it will not die from too much water leaving the cell or too much water entering the cell. We'll talk about those in a minute. So the cell is happy in an isotonic solution. A hypotonic solution is where the concentration of solute outside the cell is lower outside than the concentration of the solute inside the cell. That means there's more water outside the cell than inside the cell, so water will move into the cell in a hypotonic solution. If the cell does not have a cell membrane, then water will move into the cell membrane will expand, if the cell doesn't have a cell wall, the cell membrane will continue to expand as more water moves in until the cell membrane pops. And that will kill the cell by osmotic lysis. Another way is if you damage the cell wall with something like a penicillin or lysozyme, then the cell membrane can protrude out of the holes in the cell wall and water can move into the cell, and the cell membrane will protrude out of the hole, and then water will move further into the cell, protruding the cell membrane further out of the hole until that cell membrane will pop, and that'll kill the cell. On the other hand, if a cell has a cell wall, like most bacteria, and the cell is in a hypotonic solution, and water moves into the cell membrane can expand up until the point where it touches the cell wall. After it touches the cell wall, the cell membrane cannot expand any further. It's prevented by the cell wall. That will stop the water from moving into the cell because there's no further room for the molecule to move into the cell. Oh, it's telling me my internet connection is unstable. So if you need something repeated, just let me know. Hopefully we won't lose the connection. So the cell wall can protect the cell from osmotic lysis. Most bacteria live in a hypotonic solution. For example, when a bacteria infects us and is growing in our tissues or in our blood, it's in a hypotonic solution. And then water will move into the cell until the cell membrane reaches the cell wall. Then the cell membrane can't expand any further. It will not expand until it pops. And that's how penicillin G damages the cell. It puts a hole in the cell wall, allowing the membrane to protrude out of that hole. And then the cell can take in more water until that water pops the cell membrane. A cell can also be in a hypertonic solution. That's when the concentration of the solute outside the cell is higher than the concentration of the solute inside the cell. In this case, there's more water inside the cell, less water outside the cell. So water will move from inside the cell to outside the cell. This can result in plasmolysis, where the cell membrane will pull away from the cell wall. And generally the cell membrane will shrink, and that will lead eventually to cell death, depending on how sensitive the cell is to drying out. Many cells like E. coli are very sensitive, and so in short term, when an E. coli cell is put in a hypertonic solution, the cell will soon die, such as when E. coli falls into a bottle of molasses. Water will leave the E. coli cell, killing the cell by plasmolysis. Any question about any of that? All right. So passive processes are okay when the concentration of a substance required by the cell is higher outside the cell than inside the cell. But what about when a cell is in an environment with a low nutrient concentration? For example, a cell in the open ocean. How is it supposed to survive if the nutrients outside the cell are at low concentration? Or in the desert, like the Sahara Desert, nutrients outside the cell will be very low. So let's talk about active transport, where the molecule is moved against its concentration gradient from areas of low concentration to areas of high concentration. And when a cell is in one of these environments, or whenever a cell needs a molecule that is at a concentration lower outside the cell than inside the cell uses an active transport mechanism to move the molecule inside the cell. Molecule is moving against its concentration gradient, so it's going to require energy to move the molecule from areas of low concentration to areas of high concentration. For example, a cell in the open ocean finds a glucose molecule outside the cell. There's going to be many more glucose molecules inside the cell than outside the cell in the open ocean. And so the glucose will be actively transported against its concentration gradient and into the cell. This active transport requires energy, and the cell will get that energy it needs from ATP. It also requires an active transport protein to physically pump that molecule against its concentration gradient, in this case, from outside the membrane to inside the membrane. You have one transport protein for each molecule you want to transport into the cell, meaning each type, like glucose, will have a different active transport from lactose. And then that transport protein will require energy to pump the molecule against its concentration gradient. And one ATP molecule will be used to move one molecule into the cell against its concentration gradient. Any question about any of that? ATP is adenosine triphosphate, and we'll talk a little bit more about that. All right, so all cells can move molecules against their concentration gradient by active transport. But in prokaryotes, we have another means of moving a molecule into the cell against its concentration gradient. You can move it into the cell by active transport, but you can, in prokaryotes, also move it into the cell by group translocation. Group translocation is a special form of active transport, and this special form is only found in prokaryotes, being the bacteria and the archaea. It does require a transport protein to pump the molecule against its concentration gradient. And it does take energy to move the molecule against its concentration gradient. But in this case, the energy does not usually come from ATP. It comes from a high energy phosphate compound, such as PEP. And I can mention that ATP is a high energy phosphate compound. But my point is, it's just a non-specific high energy phosphate compound. And frequently it's more often PEP rather than ATP. In addition, the molecule that's moved into the cell is chemically altered. For example, when we move glucose from outside the cell to inside the cell against its concentration gradient using group translocation, the glucose is moved into the cell, but while it is moved into the cell, the glucose will be chemically altered from glucose to glucose 6-phosphate. And usually the phosphate group from the high energy phosphate molecule is transported onto the molecule moving into the cell. So PEP, for example, will have the phosphate group lose PEP and then be added to glucose. So glucose outside the cell be moved into the cell, but inside the cell, it won't be glucose, it'll be glucose 6-phosphate. And this transported molecule is chemically altered by a group translocation. Let me go through an example here. So let's say inside a cell, we have 10 glucose molecules. And now outside the cell, we have, let's go, just to make it simple, 10 glucose also. So the concentration is equal inside the cell to outside the cell, so there will be no net movement of glucose. We could move all of the glucose inside the cell by active transport. Now, the first one that's moved into the cell has a concentration gradient it has to move against. Will be one glucose moving into 10 So let's say, let's just say 10 to 10, but so the concentration gradient for moving the first glucose into the cell will not be very stiff, and that'll make it 9 outside the cell, and now 11 inside. The point is the concentration gradient is not that great that you're moving it against. But now let's move the 10th glucose into the cell. Let's skip down to here. Now we're moving against, it's something like 1 to 20. And there will be 0 outside the cell. Oops, didn't do that right. Can you guys see this? You want me to blow that up? And 20 inside the cell. Let's do an arrow there. Up. Let's get rid of the cell. See if I can shrink that. That's a very stiff concentration gradient. You're moving glucose. 1 outside the cell and 20 inside the cell at the end. Now, if we use group group translocation for half of them, and then active transport for the other half. Instead of having this 1 outside the cell and 20 inside the cell, the worst we will have for this will be something like, let's go with this here. The 10th glucose moved into the cell. Let me just copy all of that. The worst you'll have is 1 moving into the cell, and let's see, it'll be 15 because 5 are moving by group translocation and 5 by active transport. So instead of being 1 to 20, it'll be 1 to 15. And that's less of a concentration gradient you're moving the molecule against. And so it's thought that with group translocation, you're making it easier for the prokaryote to move molecules against their concentration gradient. Any question about any of that? Did you guys sort of understand what I'm trying to show here? And that is when you have both active transport working, you can have half the molecules being moved into the cell by active transport, like half the glucose, and then half of the glucose moved into the cell by group translocation. That makes the steepness of the concentration gradient less steep, because instead of moving all of them in by active transport, you're only moving half of them by active transport and the other half by group translocation. Any question about any of that? All right. So it's thought that group translocation allows cells to accumulate substances more easily because the cell can use both group translocation and active transport to move the molecule into the cell. And remember, when it moves it into the cell by group translocation, you no longer have glucose inside the cell because glucose outside the cell will be changed to glucose 6-phosphate. All right, let's talk a little bit about the cytoplasm. This is the substance inside the cell membrane, at least of prokaryote cells. It's mostly water, about 80%. It does have dissolved solutes in it, like glucose, amino acids, proteins. Even the DNA is usually dissolved in the cytoplasm. And I think, at least in eukaryotes, the only time the DNA is not dissolved in the cytoplasm is when the cell is actively undergoing reproduction, either mitosis or meiosis, and then the chromosomes are condensed. When the chromosomes are condensed, the DNA is not dissolved in solution, but most of the time the DNA is dissolved in the cytoplasm, at least of a prokaryote cell. It would be dissolved in the nucleoplasm of a eukaryote cell. The cytoplasm does include more than just the solutes that are dissolved in it. It does include the nucleoid region, and this is wherever the DNA happens to be. Wherever the DNA is floating around in the cytoplasm, there is no distinction between the nucleoid region and the cytoplasm because there's no barrier. It's wherever the DNA just happens to be floating around in the cytoplasm. The cytoplasm does include ribosomes and inclusions. These are molecules that are so big, or accumulations of molecules so big, that the accumulation is no longer in solution. Like a ribosome is such a big molecule, it is not in solution. And there's an inclusion right there, and those are ribosomes, which you can sort of see the ribosome dots there. That might be an inclusion right there. The nucleoid region or the nuclear area is wherever the DNA is located. It can move about in the cytoplasm, wherever it floats around. There is no membrane to separate the two. It does stain differently, which is why it's called the nucleoid region. There can be plasmids as well as the chromosome. We'll talk a little bit more about plasmids when we talk about genetics. A plasmid is just a small molecule of DNA. You can find ribosomes in the cytoplasm. They have subunits. You don't need to know the size of the subunits. You do need to know that the ribosomes are the site of protein synthesis, and that the prokaryote ribosomes are smaller than eukaryote ribosomes. The prokaryote ribosomes are 70S in size. The eukaryote ribosomes are 80S in size. You should know that ribosomes are made up of both protein and RNA. So there's ribosomal proteins and ribosomal RNA that make up ribosomes. Two molecules make up ribosomes. Ribosomal RNA, also called rRNA, and then ribosomal proteins. Any question about ribosomes? All right, let's talk a little bit about inclusions. Inclusions are something the cell is accumulating inside the cell. And initially, at low concentrations, the material will be dissolved in the cytoplasm. The trouble is the cell will accumulate more of this material until eventually there's so much of this material that the material precipitates out of solution. And once it precipitates out of solution, we will call it an inclusion. You can actually see an inclusion under the light microscope because it's this blob of stuff that has been accumulated in the cell, and it comes out of solution, so then you can see it. If you ever worked with chemistry earlier and you were putting a bunch of salt in solution, and initially the salt will be dissolved in solution, but then you put too much salt in solution, and then a whole bunch of salt will just crystallize. That's because the salt has precipitated out of solution, and that's what happens when we have an inclusion form in a cell. Initially, we have the material dissolved in solution, the cytoplasm, but then we accumulate too much of it and it can't be held in solution. And so it precipitates out of solution, forming the inclusion. You do need to know there are different types of inclusions, but you do not need to know the types of inclusions. Does that make sense? You only need to know there are different types. Inclusions generally are reserved deposit within the cytoplasm, and the cell will use these when resources are low. Usually, not always, but that's usually the case. For example, this one here, this is a cell that can metabolize sulfur. So this cell will actively bring sulfur molecules into the cell. And initially, when the sulfur is at low concentration inside the cell, the sulfur will be dissolved in the cytoplasm. But after the cell accumulates so much sulfur, there's too much sulfur to be held in solution, and the sulfur will precipitate out, causing the inclusions that we see here. And they're called sulfur granules or sulfur inclusions. Here are some other inclusions, magnetosomes, and let me see if I can blow this one up. Right there in yellow, you can see the cell, cytoplasm, and there are three inclusions, the metachromatic granules. Any question about inclusions? All right. Let's move on to our last thing for today, and that is the endospores. Endospores are a specialized resting cell produced by certain species, mainly members of Bacillus and Clostridium family. These specialized resting cells are highly resistant to desiccation, heat, chemicals, radiation, and desiccation. They can survive conditions that the parent cell, called the vegetative cell, cannot survive. So the endospore is a special structure that is allowing the cells to survive in harsh environmental conditions. And then when the conditions become more favorable, the endospore can germinate and return to a vegetative state. The vegetative state is the normal metabolically active cell that can grow and metabolize and do everything the cell can normally do. The endospore is a resting cell, which it does have some metabolism, but essentially none, meaning metabolism is very low in the endospore. Endospore formation is called sporulation or sporogenesis, where the endospore is forming. And here, let me blow this up. You can see these cells are forming endospores because the environment is deteriorating, and the parent cell, like this one here, which doesn't have an endospore, knows it's going to die, so it begins making an endospore. And there's an endospore inside a parent cell, and the same with these right here. So the parent cell makes an endospore, and then the parent cell will die. One cell makes one endospore, that's one makes one, that's not reproduction. This is a survival strategy, not a reproductive strategy. Any question about any of that? If not, I'm going to stop here, and I'll see you in the lab at 6:30. Any questions? All right, if there's no questions, I'm going to say goodbye.